Well, welcome everyone. As the director of this institute, it's my pleasure to welcome you. I hope you've been having a good course so far. Yeah, they're taking care of you okay? Everyone's treating you well? Okay, that's good to hear. So what I'd like to do today is talk about our research on honeybees and how it gave us a very important lesson about the relationship between genes and behavior in general. So the lesson goes beyond the confines of the beehive and is kind of an interesting example of how work on model systems and basic biology can yield insights that can then go into the direction and become more applied. So I'll tell you a story about a discovery that we made in honeybees that has widespread applicability to all organisms, especially including humans, and how the work in humans is just starting to take off. So with that as the context, I will frame the honeybee story in a larger context related to just general aspects of genes and behavior, and how we think about these right now in the context of humans. So where we are right now, in my opinion, is kind of summed up in this slide here. And that is, for certain kinds of behaviors, we really have no problem talking about the influence of genes on those behaviors. So sports, it's fine to look at a family like the Mannings and just think about, the apple doesn't fall from the tree, and we see that picture and it makes perfect sense, and we're totally comfortable with that. By contrast, other behaviors, such as intelligence, such as certain personality traits, epitomized by that picture below, we're less comfortable with. We are uncertain about the relationship between genes to these particular behavioral traits, and perhaps more importantly, we're really concerned about the social and political, economic implications of such work. After all, as I tell my students, next to nuclear physics, the field of behavior genetics has done more harm to people than any other part of science. So those of us that work in this field have a legacy of shame to live down and owe it to everyone to get it right this time. So we are very concerned about the relationships between genes and behavior, and at this moment, at this point in time, we have this dichotomy. We're worried about genetic determinism and the implications of that as they have played out horribly in history, and as they could play out again without the proper consideration of the science. So right now we have this feeling still that it's nature versus nurture, that it's genes versus the environment, all or nothing, apples or oranges, whatever kind of dichotomy you want to paint, that they are very different entities. And we're uncomfortable with how to integrate them, both scientifically and, as I said, in a larger context. So what I want to tell you today is how honeybees have solved the nature-nurture problem. How we have used honeybees to make a discovery that gave us new insights that have widespread applicability beyond the beehive. So more specifically, how we used honeybees and genomics to solve the nature-nurture problem. And in doing so, discovered a new property of the genome, its dynamic property, the property that allows the genome to respond to the environment. So I want to just set this up in the context of behavior, remind you of some basic relationships between DNA, RNA, and protein, but also put this in a context of behavior. And here you see this sort of ladder of complexity or gradient of complexity with respect to behavior, where we start with DNA, we move to RNA, to proteins, neurons, brain circuits, brain regions, and behavior. And what we'll be talking about today primarily is the relationship between gene expression and behavior. Gene expression, of course, is a manifestation of RNA abundance. So those are synonymous. RNA abundance, messenger RNA abundance is the technical term, and more generally we can talk about that as gene expression, or even in a more general term, one can say gene activity. So the essential concepts, I think you are all familiar with these and have gone over them already in the class, but just very quickly, we're talking now about the central dogma, the trinity. DNA is transcribed into RNA, and RNA is translated into protein. So we're going to be talking about this intermediate step here, the transcription of DNA to RNA. RNA then, of course, is translated to make proteins that build the nervous system, that run the nervous system, and so forth. But we will be measuring RNA. Just as an aside, if we could measure proteins, all the proteins that are expressed in the brain with the same facility that we can for RNA, a lot of the work would be done on proteins instead of RNA because it's closer to the business end, one step closer. But proteomics as a field, measuring all proteins, still lags behind transcriptomics, which is the measurement of all genes. And so as scientists are wont to do, we look under the lamppost for the keys. It's a better place to look. Yes?
Why is it the case that proteomics lags?
Proteins undergo many more modifications. They are so much more different from each other than RNA. And so you have to incorporate so many more analytical tools to be able to describe and then measure them quantitatively. So the central dogma figures into this. You folks are well familiar with that, I'm sure, and we're focusing on it at the RNA level. And then, of course, the difference between genomes and genes. So we're going to be talking mostly about genome-wide signatures, looking at the gene expression activity across all genes. And then, of course, one can bore down more deeply and focus on individual genes. But the genome is the large piece of DNA found in every cell, and it's composed, of course, of all the genes, other DNA segments that regulate the genes, and then other parts of the genome that are no longer functional. So this is taking place, of course, in the nucleus. When we talk about transcription, the DNA resides in the nucleus and then it's transcribed. Those transcripts leave the nucleus and orchestrate protein. And so there are just some diagrams to make sure that we're all on the same page this early in the morning. Gene to protein, the transcriptional process. This is the last time we're going to be talking or looking at these things in any detail. From now on, we're just going to step back and talk about the process in general. And then finally, you have the genome packaged in the chromosomes. You have the intergenic regions that are seen right here. Many of these regions contain the sequences that regulate the genes, turn them on or turn them off. Okay, so with that as a primer, let's move on. So why are bees useful for this endeavor? There's two reasons. First of all, they're big enough, this may be a surprise to you, they're big enough to be able to study them as individuals. Here you see a bee with a colored number tag on it. It's possible to put identification markings on individual bees and then follow them throughout their lives. This actually is a very old slide using a colored numbered plastic tag. We now have graduated to more automated methods of monitoring bees. We use RFID tags. We also use barcodes. A paper on the first use of the RFID tags authored by Claudia Lutz and a key author, Paul Tenzer, a citizen scientist in the lab, is coming out in the next couple of weeks using the barcodes and using the RFID tags. But the point for today is that they're big enough to be able to monitor on an individual basis. They also, by the way, are interesting enough to monitor on an individual basis. They have very consistent patterns of behavior. They engage in a number of different activities that change as they get older. They have different personalities. Some work harder, some work less. Some are more likely to do one job. Some are more likely to do another job. So they have a very rich behavior that is open to very detailed examination because of these methods. And then on the right, you see part of their social life, which indicates that they live in a beehive. These white stacks here that you see here, each one of these is a beehive. It's a family. It's composed of between 40 and 50,000 individuals at the height of the season. And that's a social unit. It's not an aggregation where they just happen to be there hanging out. That's their family. It's a tightly integrated group. And so because of that, the social environment of a honeybee can be manipulated with great precision. So we are still at the very early stages of genetic manipulations, making transgenic bees, GMBs, I guess we would call them. But, and that is picking up steam. But we are very good already at altering and making designer colonies, changing the social environment of the colony. So we can make big colonies, little colonies, genetically diverse, genetically narrow. We can treat the colonies with neuroactive substances. We can do all sorts of things at the colony level, replicate it at the colony level, and then look at the effects of the social environment on behavior, and as I'll talk with you about today, on brain gene expression. So basically we're able to do social engineering on a grand scale, and for those of you who know the intricacies of human science, we don't have to fill out any forms to do that. So I'll tell you first about some work that relates to the effects of changes in the age demography of a colony and how we can trace those effects. So let me give you a little background. I mentioned this in passing, but let me make it more specific. So the adult worker honeybee shows a very intricate pattern of behavioral maturation. It changes what it does as it grows up. An adult honeybee lives about 6 weeks. It spends the first 3 weeks working inside the beehive, fancifully depicted over here like this. And then it spends the final 3 weeks, up to 3 weeks of its adult life, foraging for nectar and pollen on flowers. So that's a very predictable pattern of behavioral maturation. The young ones work in the hive, and then they graduate and become foragers. So you see a bee on a flower, that's an older bee. And if you then sat back and thought, yeah, that makes sense. Insects are little robots. They just, they can do lots of things, but they do them in very specific, invariant ways. You would be forgiven, but you would be wrong. They actually show a great deal of plasticity. They can change their behavior, and not only change their behavior, but change their behavior in response to the needs of the colony. They're able to sense the needs of the colony, change their behavior appropriately. So, for example, if you remove all the old bees in the colony, no foragers, all the old bees, some of the young ones speed up their maturation and become precocious foragers. It's like this cartoon here where this driver is only 7 years old. So you remove all the individuals in a human society of driving age, some of the young ones take up that slack and become drivers. Kind of a scary thought for those of us that have had or have young children. But that's the idea. So they become precocious foragers. We were interested to know, like, what's going on? How are they able to sense the changes and change their behavior and become precocious foragers? How do they know what to do? Question is interesting because there's a contrast between the bee society and the human society in this particular context. Human societies are known for central organization, centralized organization. There are individuals that are in charge. There's a hierarchy. So if a human being wants to know what job needs to be done, or to put it in another way, an ambitious young undergraduate is looking for what would be a great career, looking for labor opportunities, openings in the labor market, that individual will go to sources of information that have been centralized, like the Wall Street Journal, giving information, reading a column saying, oh, computer science will be a hot area to go into. That was an amount of information that was collected broadly, processed in a centralized way, and flowed into a single dissemination point. Or if you think about various authority lines, you all deal with principles. We don't have to talk about your principle or anything today, but you deal with centralized control in your work life, and that's what humans are known to. Well, honeybees, the lights are on, but no one's home. So they're changing their behavior, they're responding adaptively to changes as they need to, but no one's in charge. The queen's not in charge. The queen, unfortunately, was named in a time of human history when we were, as a people, very enamored of royalty. If she were to be named today by perhaps some of the more disrespectful students in your classes, she would get no better name than, yo, egg-laying mama. And that's really all she deserves in this context, because she doesn't tell the workers what to do. She has her job, she lays eggs, she does it very well, thank you, 2,000 eggs a day for up to 2 years, but she doesn't tell the workers what to do. So how does an individual worker bee know what to do? That's the context. So what we found, to cut to the chase, is that there's a process of social inhibition that goes on. Old bees inhibit the maturation of young bees. Old bees inhibit the maturation of young bees. The more old bees are present in the colony, the slower is the rate of maturation of the young bees. You remove the old bees, you're removing the source of inhibition, and so then you have premature maturation. I want to tell you now a little story about how we made this discovery. And the moral of the story is that serendipity sometimes really does help. And that's the story here. So once upon a time, 24 years ago, I was a young assistant professor in my first summer here. And we were just starting to address this question of how do bees know that there's a shortage of foragers or that something's wrong with their hive and they need to become precocious foragers. So the way we had planned to do this was with the very first postdoc in the lab, Professor Zachary Huang, now an associate professor at Michigan State. The way we decided to address this question was we made a glass wall observation hive so you could see into it like this picture has here. We put colored numbered tags like you saw before on a bunch of the bees, and we created a colony without any foragers. And then our plan was to watch the tagged bees and let them grow up. We assumed some would become precocious foragers, some would not. Only about 10 to 20% of a colony become a precocious forager. They always maintain a division of labor, some working in the hive, some foraging. So we thought we would get long-term behavioral profiles on these individuals, some becoming foragers, some not, and then pore over the records, pore over all of those observations that were made during that period of time, hours and hours over the course of a week. They didn't even call it big data back then, but that's what it was. And then see if we could pick out the differences between the individuals that became precocious foragers and those that did not, right? Pretty straightforward, kind of watch the process unfold. So that was the plan. Meanwhile, Hollywood called. Actually, it was Pittsburgh, but it sounds a lot better to say Hollywood. A producer of a public PBS show was calling. They were making a show on a documentary on social insects, and they wanted to come and film my lab. So that was, of course, very exciting. Of course, I said they could. They came, and they needed an observation hive to film the work. No problem, we've got an observation hive. It even has tagged bees, and everything should be fine. So they looked at the observation hive. They said, no, it's too small. We need another observation hive. You know how those types are. There's nothing is good enough for them. So we had a little bit of a difficulty. The old bee lab, the very first bee lab that my lab was given here at Illinois, was a lovely facility, but it was a little small. The place where the observation hives were located, we had space for 2, but they were really close together. And that's a problem because if beehives are too close together, some of the bees can go into the wrong hive by accident. Now, don't get me wrong, bees are brilliant, they have amazing capabilities, but they did not evolve under high density conditions like that. They evolved where beehives were maybe every 500 meters or so. So they can find where they live really well, but if you pack them in too closely, some can drift in. So I was a little worried about doing that, worried about maybe screwing up Zachary's experiment of having the wrong bees go into the hive. We didn't know what that would do, and was a little worried, but you know, I didn't worry about it that long because it was Hollywood, you know, Pittsburgh. They were a PBS show, you know, they wanted us to be on it. So of course we gave them the observation hive. They filmed it, everything was fine, I was in it, my mother was very proud, and I thought we were all set. But a couple days later I asked Zachary, well, how did the experiment, how's it going? He said, well, actually not very well. Bees from your PBS hive drifted into my little hive, and there's no foraging at all. None of the bees in my hive were foraging. So I felt terrible, guilt-ridden, you know, the pursuit of vanity and glory ruined the experiment of my postdoc. But then I had a little insight. and that is maybe all is not lost. So there are two classical ways that you approach biological experimentation. One is you do a kind of a naturalistic approach. You're studying process X, whatever it is. You create process X, you let process X unfold, and you measure it in various ways. That's what we were doing. We wanted to see what's regulating precocious foraging, so we staged a situation to have precocious foraging, watched it unfold, and then hoped to be able to get the insights as to what makes precocious foraging tick. That's just one way to do biology. The other way to do biology is to take the process that you're interested in and break it, and then see how you've broken it and get insights there. That is probably the dominant mode of science, of biological science at this point in time, characterized best by the mutant approach. You want to know how learning and memory works, you create a fly or a mouse that doesn't learn or remember and see what genes you have affected, and so on and so forth. So I thought, okay, well, Zachary said that bees drifted in and now there's no foraging on the part of the resident bees. Maybe we broke the very process that we want to study, precocious foraging. Maybe there's, rather than looking for what's promoting precocious foraging, which is what our dominant mindset was, maybe there's something that inhibits precocious foraging. And we accidentally did that by having the bees drift in from the larger hive to the smaller hive. Does that make sense? So here we have the small hive precocious foraging. Here on the left we have the PBS film crew hive, and on the right our experimental hive. What happened, just to recount, is that in the pursuit of vanity, I allowed bees from the PBS film crew hive to drift in to the little hive, and as a result we had no precocious foraging. So the hypothesis based on this anecdote, because I'm sure you teach your students you can't believe an anecdote, the hypothesis based on the anecdote is that old bees inhibit the maturation of young bees. We then proceeded to stage experiments that specifically tested that. We took old bees, transplanted them in precise numbers into our experimental colony. We transplanted young bees as a negative control. We transplanted different numbers of foragers and so on and so forth, and established that that's in fact exactly what's going on, that there was this process of inhibition. And then what we found was that this process of inhibition is associated with changes in the expression of genes, that precocious foragers look like normal foragers. They have a pattern of gene expression that resembles the forager rather than bees that are the same age. So we got this first inkling of a dynamic genome, that the changes, that differences in gene expression, that gene expression is not static, that it depends on the environment. And you take a bee that's developing under normal conditions, where when it's young it works in the hive, and when it's older it becomes a forager. You then compare that to a situation where you have a lack of social inhibition and some of the bees are growing up faster. Those bees show a different pattern of gene expression than their same age sisters. They now look more like foragers than nurses. So their gene expression profile is not fixed. So we got our first inkling of that, and now in the second project and the final project that I'll talk with you about, we were able to see this even more clearly. So I just told you about social engineering having to do with manipulating the age demography, changing the age structure of a hive, having in this case small hives with only young bees. Now I'm going to tell you about a cross-fostering experiment. Before moving on, I'll just see any questions you have on the first story. Yes?
How big does a colony have to be before you can actually run an experiment on it? So you said it reaches up to 40,000, right?
Yep. We did some experiments on this project with the world's smallest beehive with one bee in a plastic dish. And we found that those bees, that was another way that we got insight after we did this transplant experiment, that it's an inhibitory process. So one bee in a dish for 1 week in the laboratory matures rapidly because there's no inhibition. When we put it in a hive, she starts to become a forager. Yes?
How quick does that maturation occur? Is it a week? Is it a day? Is it, if the bee is maybe 5 days old, how quickly
Mm-hmm.
does it turn into a forager?
We see it within a week, so we measure it after a week of exposure to this or that. Precocious foraging can occur as early as 4 days. If you throw a couple of factors at them, starve them, and put them in a colony without old bees, they can start as young as 4 days. We presume that there are indicators of that happening, changing sooner. Changes in gene expression, changes in physiology, changes in muscles, wing muscles especially. But all those measures are destructive, and so we can't see them early on. Yes?
Do you see any significant differences between male and female bees?
Yep, there are big differences. So all the bees that I've been talking about are female. They're worker bees. In the honeybee society and in their cousins, the ants and the wasps, the males have very distinct roles that have nothing to do with colony growth and development. The only known function of the males is to mate with virgin queens, and they do not engage in any other colony activities. Yes?
Does the cell cycle actually speed up as well, or how does maturation?
Yeah, that's a great question. That's not been looked at. There are some hints that the circadian clock is affected, and so that now we know that the circadian clock is related to the cell cycle. So that's a possibility, but that hasn't been it hasn't been looked at. Anything else? Okay, so then the second story then has to do not with behavioral maturation, but with aggression. And it has to do with another social engineering technique, which is to cross-foster. Cross-fostering means to take individuals from one location, put them in another, and vice versa. And so there's some classic experiments in genetics with common garden experiments, moving plants from one environment to another, and vice versa, and so forth. So we can do this with bees, put them in one hive, residents in one hive, and move the residents from the one hive into another. It has to be done with precaution. You can only do that for very young bees. Otherwise, they will fight when they get in the strange hive. But for very young bees, one can do that. So we looked at the dynamic genome, the idea that the environment is affecting gene expression, which was the first hint we got from the precocious foraging story. We looked at that in the context of aggression. So let me introduce bee aggression to you before going into the details of the story. So bee aggression is very complicated. It's a very intricate, sophisticated system, and involves hereditary, behavioral, and social components, which I'll explain to you in a moment. But first, let me give you the ecological context. The ecological context is as follows. Bees live on flowers, as you know. They make a living off of flowers, what I mean. They collect nectar and pollen. Nectar and pollen, let's just focus on nectar. Nectar is present in individual units in flowers in really, really tiny quantities, microliter quantities. Furthermore, nectar as a sugar source is, okay. It's not great. It's between 10 and 40% sugar. What the bees do is they collect with hundreds of thousands of trips collectively, like the distance around the world, those kinds of numbers, right? You read on the honey label. Hundreds of thousands of trips. Collectively, they take this resource, which as a resource is so-so. It's distributed patchily in space, a few flowers here, a few flowers there, within each flower a tiny amount. It's distributed patchily in time. Today's bloom, tomorrow it's gone. And they take that, and by dint of all of their hard work as individuals and all of the incredible social organization of the hive, they turn that into a bountiful resource called honey. And they have pounds of it, tens of pounds of it, 50, 70, 100 pounds of it, depending on the part of the world that they live in. Honey is not nectar. It's been transformed from nectar. It's about 85% sugar. It's preserved, so it lasts for a long time. And it's all there in one place. Now I ask you, if you're Winnie the Pooh and you have a sweet tooth, what would you rather do? Go from flower to flower and get like a microliter of 40% syrup, nectar, or knock over a beehive and have like 50, 80, 90 pounds? this is a long-winded way of saying that the very traits that make honeybees successful also have made them vulnerable. They concentrate resources to one place, making them very attractive to a variety of animals, little animals, medium-sized animals, big animals, as a source of food. And that's just the carbohydrate side. I could wax similarly about the protein side, but I'll give you a break. So you get the idea. So because of that, bees then have evolved very intricate defensive systems to defend the resources that they collect and work on and store. So this system then involves hereditary, behavioral, and social components. Hereditary components, genetic components is what I mean here, is that bees that live in parts of the world where they are more vulnerable, where there are more predators, will be more aggressive, will have a more active defense of their hive. I do want to say, we'll be talking about aggression, but just for the record, honeybees deploy their aggression only in defense of their hive. They are not aggressive creatures per se. They're not out hunting in any context at all. It's purely for defense. So they will mount a more vigorous defense, and there are genetic differences. There are strains of bees that are more aggressive. So if you're a hobby beekeeper, you try to buy gentle bees and keep them in that context. There are behavioral components to aggression. So I mentioned before that different bees do different jobs. We talked about working in the hive or foraging. The context of aggression is also, you see this kind of specialization. There are two groups of bees in particular. One group is seen in these pictures here. These are guard bees that patrol the entrance. Here's one right here with her front legs off the substrate, her antennae forward, her wings partially back, which you can't see. These bees patrol the entrance like bouncers in a bar. They can distinguish individuals that belong from individuals that don't. And that even includes conspecifics, other honeybees. Yes?
So then, when you were talking about having to transplant foragers into a hive previous experiment, was there issues with certain bees not accepting those foragers?
There would be if we didn't take precautions. So the trick with all of that is working with very young bees. Very young bees, as residents, are not yet able to do these kinds of distinguishing abilities, and so they will accept other bees. So, yes?
So both sets of bees need to be young, the bees that are accepting and the bees that are coming in? Or
You can have one or the other depending on the experiment. So we were able to transplant foragers into an all-young bee colony.
Okay.
And if we want to take bees and put them into a colony that has old bees, the transplanted bees need to be young. So you need one part, the equation has to have one group young.
That's interesting that the young ones could be transplanted into an older hive, though, because the older hive presumably would have bouncers.
Yep, they would. Yeah, in that case, the young bees don't really have a complete set of recognition characteristics.
Okay.
Bees, like many insects, rely heavily on olfaction, and so they smell each other, and the young bees don't have a complete recognition smell yet, and so they
The
can...
old bees can't.
Right. Yes. Question?
I was actually wondering about the olfactory.
Yeah. One more here? Yes.
So each hive will have its own kind of set of chemicals that is distinct, that the guard ones can tell from another?
Yep. Yep. And like the theme of this entire talk today, it's a combination of inherited and environmentally acquired. So, for example, there's a famous flow of honey in England, heather honey. Have you ever had heather honey before? It's very distinctive honey. And the nectar of heather is really strong smelling. So if you have a bunch of beehives that are all collecting heather nectar, you can put bees from one hive into the other. They can't tell the difference. They all smell like, it's like going after an Italian restaurant. Everyone smells like garlic, So they all smell like heather. They can't tell who's who. But if you have a situation where you don't have such an overriding environmental chemical, then the intrinsic, endogenously produced self-chemicals are more predominant. So you've got one group of bees, the guards. They are the sentinels. They are the ones that patrol the entrance. When they detect a foreigner, they react by releasing a social signal, an alarm pheromone, a chemical, that alerts the other bees in the hive. The group that it alerts the most, the group that it has the strongest effect on, are the soldiers. That's the second group. Soldiers are old bees. They're foraging age, but they seem to either forage very little or not at all. They hang around the hive. They're the first bees that respond to the alarm chemical. They respond by flying out, looking for an intruder to sting. So you've got guards and soldiers. They are coordinated in their activity by an alarm chemical. So these are the components of the aggression system in the bees. So what we did is we took advantage of these heritable differences, and we took advantage of, I'm sure you've all heard of the famous African bees, the killer bees, that are much more aggressive. They are not located here, for the record. They are in Mexico. And so we did an experiment where we took European bees from hives that are derived originally from European honeybees that came over to America several hundreds of years ago. And those bees have a relatively gentler disposition relative to African bees. And we did the cross-fostering experiment, and we compared European bees in African colonies, European bees in European colonies, African bees in African colonies, and African bees in European colonies. A 2x2 complete cross-fostering design. What we found, and then we measured brain gene expression. We collected guards, we collected soldiers, and we looked at their brain gene expression. We found big differences between European bees in European hives, African bees in African hives, as we might have expected, knowing such big differences in their behavior. But, and to the point for today, the cross-fostering showed also big differences. So when we did a statistical analysis and we asked, okay, what's the effect of the bee's individual identity? What's the effect of the colony that the bee lives in? We found 25% of the differences in brain gene expression are just due to the hive, to the social environment. European bee tends to look more like an African bee if it's in an African colony, and vice versa. So that then was a very strong finding about the dynamic genome, that just putting a bee in a different hive for a period of time, in this case for a week, leads to changes in gene expression. We are actively studying this process. So what is it about, say, the aggressive environment that makes a bee's brain gene expression profile change? What's going on? Are there sounds? Are there smells? Are there touches? How does a bee get that information? This is now we're sort of broadening the scope here as we finish up. This is sort of getting to the big question. So a big question in human biology now is, how does social environment get under the skin? We are aware of all sorts of effects of trauma, of long-term stress, of differences in socioeconomic status, of disease. All sorts of factors can have long-term effects on health and wellness. And the question is, how does that, if those are from the environment, how do they get under the skin to change human biology on an enduring basis? The question is of more than academic interest, because the idea would be, if we knew how stress causes these changes, gets under the skin, we could maybe try to ameliorate those effects. So the dynamic genome discovery in the honeybee first led to seeing this on a very widespread basis across animals. This is not just in honeybees. Many animals show this dynamic genome response where the environment affects gene expression in birds, in other social insects, in rodents, in fish. This is a very active area now of research. Yes?
Take the bees out of the hive, do they?
If you take like the European bees out of the African, put them back in the European, we've never done that before. That would be interesting to see. The guess would be yes. Now we're limited by being able to measure brain gene expression only once, but we could have the right controls to infer that. So the dynamic genome is alive and well, and it's widespread in the animal world. And what we now have is a new model for for the relationship between genes and behavior. We have social information somehow getting under the skin, affecting the genome. So as I remind my students, the genome doesn't, it's inside the nucleus of a cell, right? So it doesn't have ears, it doesn't have eyes, it doesn't smell. So how is the information getting there? So you have to imagine, depending on the behavior and the animal and so forth, you have to imagine, or the environment, you have to imagine a set of sort of transduction events whereby environmental stimuli are transduced, they're perceived by the animal, they're transduced into various internal signals that finally reach the nucleus to cause changes in gene expression. It's a really active area of research. Epigenetics is kind of generally how it's referred to. And at its heart, the question is, how does information from the environment get under the skin? So somehow it gets under the skin, it affects the genome, and then it changes brain and behavior. So I neglected to mention that when you cross-foster these bees, they take on partially the characteristics of their new residents. So European bees will become more aggressive if they are in African colonies. African bees become less aggressive if they are in European colonies.
And
Yes?
is that measured with brain expression? Are we talking about like not just they don't show that behavior, but we've actually been able to measure, or is that strictly behavioral?
Gene expression also, yeah. So gene expression is,
So it is physiological.
yes. So when you, oops. Okay. So when you do the cross-fostering, you will then be measuring, after that you measure changes in gene expression, and that is what you can measure. Then you do the analysis of variance, and that's how we got the 25% figure. So 25% of the differences in gene expression are attributable to location. So we're now going to have a review of everything that I said. As teachers, you'll love this. Yes?
I was just wondering, so you mentioned that the guard bees actually went out and they stung other attacker bees, or, okay.
Yes.
And what happened as a result?
So guard bees usually are the ones that release alarm pheromone, and then the soldiers are the ones that will go out and sting.
And kill?
They attempt to. It depends on how it goes. If it's another bee, then they often aren't able to kill the other bee because they can pull their stinger out, and sometimes they don't get a lot of venom in, but that is the quote intent. If it's a large animal, the venom is not enough to kill. That's sort of an aside. It seems as though honeybee venom, if you look at it, most of the chemicals cause pain rather than kill cells. And my own opinion is that they have evolved a neat trick where their bark is worse than their bite. They play on our cognitive sense of pain as an honest signal, when in this case the pain is not an honest signal. But that's a whole other story. Yes? Yeah, sure, go ahead.
Are the honeybees, or I've heard that some bees actually die when they sting, and then others are fine and can do it again?
So it depends on who they're stinging. So they have, all honeybees have barbed stings, and if they're stinging a fleshy vertebrate like us or Winnie the Pooh, the sting stays in, they can't pull it out, and so they die. If they're stinging an invertebrate, it's possible, because it's way less fleshy, to be able to pull it out.
And
then they can sting, but
they can also make more of the venom?
they can generate more venom, yeah. It's not clear how long it takes them to do that. And it's not clear under those circumstances if they're releasing all of their venom, because usually they release all their venom when the sting stays in, the gland is attached to it, and it just pumps in, as it's like a really great kamikaze behavior. Yeah. Yes?
So when the bees are cross-fostering in a new hive and they're recognized by the new hive, are they no longer recognized by their own hive, and are they permanently recognized by the new hive?
They would not be able to go back and forth. After they assume the colony odor of their adopted hive, that odor would be different than their resident hive under most circumstances, unless it's the Heather story.
Yeah.
Yes?
I just heard about beetles, that some beetles parasitize bees and they get by the guards.
Mm-hmm.
But with hives each having their own chemical signature,
Mm-hmm.
how do beetles just play with a totally different tune?
Yeah, that's a cool question. It points out the limitations of a process that has an environmental component to it. So what they do, this is probably better known in ants. Some of these beetles sneak in, and they avoid detection initially, and then they wait for nature to happen, which is they wait for the odors of their colony to start getting on their little hairs and soak into their skin, as it were, their cuticle. Now they smell like everyone else. And because of that plasticity, ant can't tell the difference. So it's, in bees we see the same thing. So I mentioned in passing that a queen will lay 2,000 eggs a day for up to 2 years. Sometimes queens will fail, or sometimes beekeepers want to replace a queen with a gentler stock, and so on and so forth. And you might say, well, how could that happen? How can you take mom away from 30,000 individuals? Aren't they going to put up a fight? And the answer is yes, they would. If you took out mom and put another queen in, a foreign queen in, and say, okay, you're on your own, they'd kill her right away. They'd say, that's not mom, and they would kill her right away. However, if you put her in a little protective cage where they can't kill her, you put a few bees from her original hive in to take care of her during those few days, then the bees in the hive are like, wow, where's mom, and well, the memory of mom is fading. Then you open up that little cage, mom! They have forgotten, and they rehabituate. Now they habituate and learn a new one. Because learning is involved, you can sometimes trick learning. So it's the same sort of thing. Okay, so this was the one that I wanted to show you. 25% of the differences in brain gene expression are due to the social environment. So we measured gene expression and behavior. So this is the old view, which we started with today, and it's alive and well, by the way. There certainly is a deterministic relationship in some contexts between genetic variation and behavior. We're struggling to find strong signals of this in different contexts, but there are good examples of it. So this is a paradigm that is fine. Variation in genes leads to variation in RNA and protein, variation in some brain systems, and then ultimately variation in behavior. So this is the old view. It's alive and well. But the new view, which we talked about today, is also alive and well, which goes the opposite direction. So it starts with the environment, with social information. There's some series of transduction, neural transduction events that occur to ultimately reach the genome and cause changes in gene activity that then change brain and behavior. So we now have two views of the relationship between genes and behavior, two arrows going in different directions, and both are right. I want to stress that, both are right. The new one gives us a lot more traction, especially for studies of human social behavior. Because what we have seen is that the original model, which is seen up on the top, the green arrow, basically was rejected by social scientists many years ago as being too deterministic, didn't fit with their understanding of behavior. And as a result, we have a very sad situation where the study of behavior now is very dichotomized. So social scientists study behavior, and I'm not talking about human behavior, talk about behavior with very little input and interest in biology. And then the biologists are studying behavior with little of the sophistication of a behavioral analysis that social scientists bring to the table. So one of the things we're trying to do here at the Institute is bring those two together based on the finding that there is a dynamic genome, that social scientists don't have to like hold their nose and do biology with a paradigm that they know doesn't fit behavior, this strict deterministic model. There is another model where their insights about the role of the environment are front and center, but what's different now, what's new now, is that those influences of the environment get under the skin to affect biology via orchestrating and changing gene expression. Yes?
If you're trying to compare two model organisms, the first arrow I'm looking at, would you identify that, like with the honeybees or the fruit flies, they have the PER2 gene. So would you look for that same gene in another organism and think that they might behave similarly. And then the contrast would be you look at a behavior and expect to find that same behavior in another organism, and then maybe you find the genes that way. So do you approach it both ways?
Yep, you can approach it in both ways. Yeah, both can, you just need a toehold to be able to get in there, and both can give you that toehold. Yep. And the reason, of course, why both can give you that toehold is because there's conservation, there's evolution, and so there are some basal things that are some basic things that can be conserved, and you can look for them and grab onto them and see how they play out. Yes?
People now comparing the differences between social organisms versus more asocial or more individual, or high versus individual.
Mm-hmm. Yes, this is a very interesting line of study. We have a project going on right now where we're analyzing the genomes of 10 different species of bees. There are about 20,000 species of bees. Don't worry, we're only just talking about one today. We're almost done. But there are 20,000 species. Only a minority are social. The rest lead a solitary life. The solitary life of a bee is you emerge as an adult, your wings mature, your wing muscles mature after a couple of hours, you go outside, you mate, you then create a nest of some kind, you lay an egg or two or three, you provision that, you die, they live as orphans, as eggs, larvae, pupae, they come out, look around, they're all by themselves. They do what I just said, and life goes on. They're very successful, very important in the environment. They pollinate many crops and so forth. Out of that basic solitary lifestyle emerged sociality, where you've got beehives like bumblebees, honeybees, stingless bees, where they live together in large groups, medium-sized groups, large groups, where they dote on the young, they feed them, they take care of them, they defend the nest, all sorts of elaborations that have occurred. We're looking at the genomes of solitary and intermediate social and highly social to see what kind of clues there might be at the genetic level to the evolution. And one of the things that we have found is that if you compare the same genes, looking at the same genes, and you look at the regulatory region, remember I mentioned that there are parts of the genes, the intergenic parts that regulate the genes turning on, you see something very interesting. So there are places in these regions that are like docking sites for where master regulator genes dock. These are called transcription factors. Some of you nodding your heads, so you've heard of those. These transcription factors orchestrate the turning on and turning off of genes. They can't do their job if there aren't little docking sites for them to bind to. So for a given gene, let's take your PER2 gene, if you look at it in a solitary compared to a social species of bee, we find more docking sites for the PER2 type genes in the social species than in the solitary species. That's a genome-wide trend, highly significant statistical result. Any one gene you may not find that, but over the whole genome. So we are really excited about this finding because it suggests that gene regulation in the social species is more nuanced, more complicated. Because if you have more places for the orchestrator gene to bind, it can regulate over a greater dynamic range. That's just pure speculation. The finding is we find more docking sites. So we hope to be able to eventually elucidate those differences at the molecular level. Yes?
When you talk about bees setting up new hives, is it a certain gene that makes, or a regulation of that gene that makes certain ones more susceptible to go off and start their own hive?
We don't know about that, but we do know that a group of bees involved in that process do have distinct patterns of gene activity, and now I'm referring to scout bees. So when bees swarm, the swarming process is a two-step process. It involves leaving the hive, leaving the parental hive, their natal hive, maybe a third to half of them, so 10, 15, 20,000, depending on the time of year, usually earlier in the year, so it's maybe 5 to 10,000 bees. And then they don't go to a new location right away. They park temporarily on a bush or something outside. If you're from the Illinois area, second base at Wrigley Field happens about every 2 or 3 years. There's a swarm that lands sometimes first base, sometimes second base. So they're temporarily located there. Then what happens is scout bees will leave that location and they look for the new permanent location, because bees live in enclosed cavities. Our western honeybee lives in enclosed cavities. So they look around for holes that are about yay big, then they go inside and they check it out. They can measure the volume, they can just check other attributes. Is it high off the ground? Does it have good sun exposure? Good school for the kids? You know, they're just checking everything out. And then they come back to the swarm and they will tell the other bees what they have found by means of the dance language. Some beautiful behavioral work done by Tom Seeley at Cornell University, and then building on the work of Martin Lindauer from Germany many years ago, has elucidated this incredible communication process, a sort of a honeybee democracy, as Tom Seeley calls it. He has a wonderful book on this, where the scouts go look for different locations. They come back and they have a lively discussion about the attributes, and that all boils down to how vigorously they dance and how long they dance. Finally, the colony makes a decision and they will fly to a new location. So the scout bees are a very distinct group, and a former graduate student in the lab, Sophia Liang, has measured brain gene expression of scout bees compared to other bees the same age, and they have a very distinct pattern of activity. Yes?
And are those bees matured from a different role also, where they previously foragers or previously some other type of bee, and then because it was time for swarming, they were specifically matured in this way?
We don't know the answer to that. Like, you know, are there some... So there are highways in bee life and there are byways in bee life. So all bees, as far as we know, clean cells, the honeycomb cells. They act to feed the baby bees as nurse bees, and then they're foragers. As far as we know, all bees do that to some extent. But then there are these byways in life, like scouts, like guards, like corpse removers, that not all bees do. And we don't know, are there some early behavioral, environmental influences on them? We know that there's a genetic component, that if you have certain genotypes, you're more likely to do certain jobs, but it's not, again, in keeping with what we're talking about, it's not a strict deterministic relationship, it's an influence. So those would be very interesting to suss out. Did they have some early experiences that make them more likely to be a scout, for example? We don't know. So, yes?
I was just wondering, is swarming normal behavior of bees?
It is. It's how they reproduce.
Okay.
So individual bees don't reproduce. This is a social level, a group level reproduction. It's like fission in a bacterial colony.
How, like every year, or what?
Depends on the part of the world. So in this part of the world, it happens on average once a year, or just slightly less than once a year, frequency-wise. In the tropics, it may be 2 to 4 times a year.
Okay, cool.
Yes?
So you were saying that there are 20,000 species of bees.
Mm-hmm.
What's the percent of genetic variation between the species? How could you determine that by looking at the genome that one is a different species?
Uh-huh. So there are statistics available that do that, and I don't know them offhand. You can look at sort of the nucleotide divergence and measure them. I just don't know that offhand. But insects are old, and so that plays into it. So the 20,000 species of bees, they are separated, some of them, by over 100 million years of evolution. So for some species, the really closely related ones, you can line up their genomes at the nucleotide level. Nucleotide, ATGC, right? Others are so diverged that you have to only align at the protein level. You can't align at the nucleotide level. Yes?
Do you find that the individual bees are, the ones that have individual behavior are older than the ones that have social behavior? Are they relatively new from an evolutionary standpoint?
Let's see. Yeah, the oldest bees that are known are solitary, but that also could be a function of just what we know, the phylogeny that's available. It is thought, so sociality is fraught. It's complex. It leads to, you know, a variety of problems. It's very successful, but it also can lead to problems. And so it's possible that some lineages of social critters are extinct. So we're not sure. So one thing that really, really bothers me, that I don't know what to make of, is that the world's oldest organisms are social. Bacteria. Are you familiar with the phenomenon of quorum sensing? It's amazing. We had Bonnie Bassler here from Princeton just last week, and she was talking about the latest on quorum sensing. It has amazing videos and stuff, great stuff for classrooms, by the way. And so these are the oldest organisms, and they're social. They send out signals, and if they get enough response, then they change their behavior and become virulent and so forth. And as a social behavior snob that thinks that this is a pinnacle achievement of evolution, I'm like, oh, well, what's going on here? I don't understand. So we think that that's the case in bees, but we're not sure in terms of who's around right now. But it stands to reason that they would have to have evolved from this simpler lifestyle. And bacteria, they have a whole different set of problems to solve, and so it's not exactly applicable. Yes?
I guess going off of that, the certain species of bees that you guys study that are social, can they adapt to an environment where they could, like if you somehow manipulated them to become the asocial type and do like nesting, like I was explaining?
Yeah. That's a really cool question. There are species of bees that are facultatively eusocial, that are facultatively able to show these traits. I have a student who just came back from Panama. She has spent now two periods of time in Panama studying such a species, that under most conditions they're solitary, but under some other conditions they can become very rudimentarily social. Now, they're not going, if you have a species facultatively social, they're just poised on that razor's edge. So that means they have not yet had the time or the conditions to evolve social traits that require a stable social group. So honeybees have a dance language. That behavior only benefits the group. If you saw an individual waggling around, it'd be like, well, what's wrong with this bee? There's nothing that it could gain from doing that. But if it lives in a social context, then it can. So that's how you start that, and then you get a group, and then the group gets stabilized, and now you have the ability to have traits that are selected for at the group level. But you're exactly right. That's where it would start, would be facultatively social. And so the interest in those species is, wow, like what's the environmental factor that does it, and how does it work, and so on. So they're really fascinating. I have to drag her back from Panama every year. Yeah,
sure.
That's right. Yeah. So just to finish up, so we have these two. The genome has two personalities. Nature and nurture are both acting on the genome, if you recall the introduction, and it's not an either-or thing. Both are acting on the same substrate. So I love this quote for that reason. The horns of a dilemma are usually on the same bull. And in this case, we're talking about the genome. And so really, it's a matter of time scale. Nature, what we call nature in the context of nature-nurture, has to do with hereditary influences on the genome. These are long time scale influences from generation to generation. Nurture also acts on the genome, but on a shorter time scale. These are environmental effects that are affecting gene activity. Which genes are turned on, which genes are turned off, where, how much, and so forth. And so this paradigm now is leading us into studies of human social behavior. We know there are many of them. For you Illini fans, this is a rare sight. The Illinois fans tearing down the goalposts the year that they went to the Rose Bowl in the 90s. So we savor such images. But in addition to tearing down the goalpost as a form of social behavior, there are other behaviors, social eating, smoking, loneliness, obesity, happiness, that are known to have, from social science, known to have social control, social influence. And now in this new hybrid field, the question is, how are they affecting biology? What kind of effects on gene expression are they having? What can we learn when we look at the kinds of genes? Because up to now, I've just been talking about genes as agnostic, right? They're just, nondescript entities. But of course, each gene carries with it functions that can reveal biology. And so looking at which genes are turned on, which genes are turned off, what biology they reflect can give some really important insights. And this has led to an effort that was started a couple of years ago. I co-organized a conference at the National Academy of Sciences where we looked at, as you can see the title, biological embedding of early social adversity from fruit flies to kindergartners, exploring what the animal models teach us and how that relates. And so it was a conference that involved biologists and of all stripes, neuroscientists, geneticists, computational biologists, and then social psychologists, sociologists, and policymakers to try to explore this, this new area. And then finally, one particular implication that one might think about is how does this new perspective relate to genetic profiling? So genetic profiling was the kind of idea that was born when we thought that DNA is destiny. You know, you are who your DNA is. We now know that part of that is true, but there's this whole other part about the environmental influence. And so how does that reflect a profiling type approach? And so that's a question that policymakers will have to be addressing, and certainly is an interesting one. I've used this as a great topic of discussion for freshman discussion classes, for example. So happy to take any more questions. Thank you very much.